Okay, our second topic is microscopic structure of the compact and spongy bone, which is again the part of connective tissue known as supportive connective tissue. Objectives of this lecture will be we understand the histology of the bone, define osteogenesis, and define ossification and stripes. So we will cover all these objectives inshallah. So before we start with the bone tissue, if we see the composition of the bone, we have organic and inorganic material which is forming the bone. So chief organic constituent or the protein we have which is forming the bone is again our collagen fibers or collagen, and inorganic calcium salts is there, and combination of these two we name it as bone. So mainly these are the two major constituents, and vitamin D is essential for the absorption of this calcium. So it is just a chemical analysis of the bone. So you can find maybe a little bit different percentages, but just as a whole to identify the percentage, 33% we have organic compounds which are collagen fibers. Then inorganic fibers which are almost 67%. The major concentration is our calcium, which is 39%, and then 17% is phosphate. So these three organic compound as collagen and inorganic compound as calcium and phosphate are forming major part of the bone tissue, along with potassium which is 0.2%, sodium which is 0.7%, magnesium 0.5%, and carbonate which is 9.8%. So before we start, we just have an idea about the cells of the bone. Osteoblast, osteo again means bone and blast means forming. So these are the bone forming cells, or they are the immature baby cells of the bone, and they are bone building cells, bone repairing cells in the periosteum, and synthesize new matrix, and the process of formation of this matrix is known as osteogenesis, or formation of this is known as osteogenesis. The second type of cells, which are mature cells, are known as osteocytes. These osteoblasts, when embedded in the bone matrix, and they change into mature bone cells, and we name them as osteocytes. They get maturity there. And the third type of cells which we find in the bone is known as osteoclast. So the class term means resorption, or they are also commonly known as bone eating cells. They are giant cells as compared to osteocytes and osteoblasts, and involved in the resorption and remodeling of the bone tissue. The term remodeling means that every time the old bone is eaten by these osteoclasts and a new bone is formed by the osteoblast. So this process of bone tissue formation is known as remodeling. So this is a diagram of a long bone which is showing a cartilage towards the head, and then the shaft of the bone, and if we can see the inner side of the bone which is known as medullary cavity, and the outer side which is a compact bone. And if we see the cells, so initially the bone cells are mesenchymal in origin. So as we all know that the body is developed from zygote. Zygote divides into a different generations of cells, and ultimately a cell which is decided to form a bone or a major bony structure of the body is coming from the mesenchymal stem cells under influence of a certain genes. And then this osteogenic progenitor cells, they are formed by these mesenchymal stem cells. So these cells now decided to form the bony tissue of the body, and as these cells grow up, they change into osteoblasts, and these osteoblasts, and then finally they will become osteocytes, which are forming the major part of the bone. These osteocytes and these osteoblasts are not only present in the periosteum, but they are also present in the substance of the bone, which we are going to discuss later on. This is one type of the cells, or one type of the formation, that from mesenchymal stem cells to osteoprogenitor cells, from osteoprogenitor cells to osteoblast, from osteoblast to osteocytes, and finally they will form the bone matrix. And on the other side we have another type of cells which I already we discussed as osteoclast cells. They are basically coming from granulocyte or monocyte progenitors. So they are basically migrating cells which come from other place, and then they form osteoclasts which are inactive, and then these inactive cells, when they get mature, they become active osteoclasts, and they are actually the bone resorption cells. And the outer part of the bone is lined by a connective tissue layer which is known as periosteum, and the inner side of the medullary cavity is lined by a very thin layer which is known as endosteal cell layers, which contains the osteocytes and osteoblasts. Now this is a diagram which is showing the histological diagram as well as the schematic diagram, and we can see that we have osteoblasts which are present there lining the bone tissue on the periphery, and as they are getting spaces and they are covered by a lacunar space and getting matured and they enter into the bone matrix, we name them as osteocytes. And along with the osteoblast or the lining cells, we have very giant cells which are known as osteoclasts. So there are two types of osteoclasts as we have discussed. One is inactive and other is active. So these active osteoclast cells, which we are not correctly seeing over here, they have projections or penetrating invaginations, and we name them as ruffled border. So we will see in the other slide. And if we see here, this is a layer of osteoblast on the outer side, this is the bone matrix and the major bone tissue in the middle, and in between them we have a newly formed matrix which is known as osteoid. So this osteoid is actually newly formed bone matrix, but it doesn't meet with the calcium or mineralized, so that's why we name it as osteoid, which is not a complete bone, but it is in middle with between the formation of the bone. So periosteum is a very dense fibrous membrane covering the bone on the outer aspect, contains blood vessels, so that is the main source of supply to the periphery, essential for the bone survival and bone formation, as we have discussed that the cells are forming there. And then we have an endosteal layer which is a thin layer. Another point which we are going to discuss here, that these cells which are periosteal layers, when they grow and they start to penetrate inside and increase the number of cells in the bone, so basically they are responsible, which are in the periosteum, to increase the thickness of the bone. So thickness of the bone depends upon the productivity of these periosteal cells, which are fibroblast cells, these osteoblast cells. And on the other side, the length of the bone depends upon the cartilaginous growth on the epiphyseal ends over here. So the cartilaginous plate is responsible for the increase in the length, or the epiphyseal ends are responsible for increase the length of the bone, and periosteal cells are responsible for the thickness of the bone tissue. So this is a diagram which is showing the endosteum, a fine layer, and a periosteum, a thickest layer as compared to the endosteum, and in between we can see the bony tissue along with the cartilage in some places. So before we move ahead, we just see that this is a mineralization of the bone tissue matrix, and here this is basically a complete mineralized bone, and when it starts, so you can see that this is a periosteal part. We can appreciate that we are looking this area only. So we are starting from the osteoblast, then we have osteoid, and then we have mineralized or complete matured bone. So how this process is taking place, just we have a look over here. So these are the osteoblasts which release matrix vesicles. So these are matrix vesicles which are released by them. So these matrix vesicles also having collagen fibers with them. And as we can see, the concentration of collagen fibers and these granules are more towards the inner side, and as they grow, early mineralization around vesicles is taking place, so calcium is starting to deposit around these bone matrix cells. And as you can see here, as these each granulous tissue is covered by a calcium tissue or calcium. So this calcium is surrounding these cells, increasing in number, and as we are going toward the center of the bone, these all granules surrounded by this mineralization of the structure of the calcium, they become confluent, and then at the end a thickened bone is formed. So this portion where these changes is taking place and this mineralization is taking place is known as osteoid layer. Above we have osteoblast, and when these complete mineralization takes place covered by a complete calcium salt, we name it as mineralized bone. So we have two types of bones, compact and spongy bone. As this is a diagram of the skull cap, which we can see we have a plate of bone which is known as compact, which is very densely packed cells are there. So we can see a plate like structure, and in between them we can see a sponge like structure which have bony tissues. This yellow material is bony tissue. In between them we have spaces, and these spaces are again lacunar spaces, just like the sponge. So that's why it is appearing like a sponge. We name it as spongy bone. This is another diagram showing the compact bone. We can't see any space there, very thick populated, as compared to the cancellous bone or spongy bone, which contains spaces in between. So it's just like a web or a network of fine bony tissue along with the spaces. So compact bone, which is also known as the cortical bone, its basic unit of compact bone is known as an osteon. So this osteon is mainly the structure which is forming the compact bone. These osteocytes with the lacunae arranged in concentric circles, we name them as lamella. These surrounded a central canal. Complex is called Haversian system, and where we find this compact bone, where we need weight bearing, we will find the compact bone. So now we will discuss the diagram. If we see here, this is a bone which is a long bone with the head, neck, tubercle, as well as starting of the shaft area, and we can see that in this shaft area, upper area, which is a head area, we can see that we have a spongy bone, and in the shaft area the peripheral part of the bone is known as compact bone. So if we see the compact bone, or we take a section of this area, we can see the compact bone. So first of all, this outer part, most outer part of the compact bone is formed by collagen fibers, and they make a circle around the bone, the whole around the bone they make a circle, and this circle, which is keeping all the tissue inside, material inside, is known as circumferential lamellae. So lamellae means a layer and circumferential means around. So around this compact bone we have a complete circle of collagen fibers, and we name it as circumferential lamellae. We go further inside from the circumferential lamellae or layers of collagen fibers. What we see rod-like structures. Each rod-like structure is named as osteon. Here we can see. So this is an osteon, this is another osteon, this is another osteon. So we can appreciate 4 osteon over here and over here also. And what we can see that in each osteon, in the center there is a space which is known as central canal. Here the enlarged view we can see this is transverse section for a longitudinal section for osteon, and we can see the space in the middle which is known as central canal, which contains an artery and a vein, and surrounded by a layer of fiber which is concentric lamella, and this single layer is known as lamella, and it is formed by collagen fibers. So how many lamella we can see in this picture? We can see 3 lamella, 1, 2 and 3. And what these lamella are actually, they are circular fibers of collagen, and here we can see also 1, 2 and 3, 1, 2 and 3. So these circular lamella are collagen fibers. So we have layers and layers and layers of collagen fibers surrounding this structure. So if I take this one lamella as a one page or one leaf, it is easily tearable, so I can tear it easily. But if I take a bunch of papers and then encircle them and then I want to tear it, it is very difficult to tear. So that means that these layers of collagen fibers are making the bone more stronger and stronger because of the presence of these circular collagen fibers. So there is not only one circular fiber, we have one collagen surrounded by another collagen, so multiple collagen fibers forming the osteons. So multiple osteons are present in this small part of the bone, and these multiple osteons with so much concentrated collagen fibers make the bone more powerful and stronger as compared to the spongy one. Another thing which we see here over here is that if we carefully see the lamella, there are three lamella which we can see in this diagram, 1, 2 and 3. So if we see the fiber direction of the lamella is different. The most inner lamella has a different direction as compared to the completely opposite direction of fibers in the lower one, and then below again it is opposite. So these different directions of the lamella are actually making the tissue more stronger, so they also add up the strength of the bone because of the presence of collagen fibers in different direction in each lamella of the osteon. For spongy bone, we will move ahead inshallah. So this is a diagram which is showing multiple osteons. So we can see osteons which are collagen fiber layers, and in between them we can see central canals, which is also known as Haversian system. And these central canals are connected together with another group of canals which are known as perforating canals or Walkman's canal. So here we can see again in this diagram, these vertical canals are Haversian canals which are running in the longitudinal direction of the osteons in the central canal, and in between them these collagen fibers, or osteons, they are perforated by a horizontal canals which are known as Walkman's canal or perforating canals. So again these are connecting the vasculature. So here we can see the blood vessels are entering into a bone. The area where they enter, we name it as nutrient foramina. So through nutrient foramen, when the artery and a vein, they are getting enter or coming out, here they divide into ascending branches and descending branches, as well as they are also penetrating forward through. So the whole compact bone is covered by these two types of canals which are running just like channels inside and providing nutrition, as vertical canals named as the Haversian canal and horizontal canal which are known as Walkman's canal. And this whole system of osteon with these vascular system is known as Haversian system. So we have layers of this with central canal. Along with that we can see this black flattened cells are actually the osteocytes. So here we can see also these brownish color lines of cells or chains of cells are actually the osteocytes. Now when we enlarge the tissue, and we can see this is the same diagram showing the osteons with the central canal and Haversian canals, and here we can see, if we see a single osteon broader view, what we can see that in each lamella or collagen fibers we have lacunae spaces, and these lacunae spaces contains osteocytes. So these are lacunae spaces, and if we enlarge a single lacunae space, and what we can see that this light color is, cream color is collagen fibers. In them we have lacunae space. Inside the lacunae space we have the osteocyte, and these osteocytes are secreting material, and we can see that they are having a processes which are known as canalicules. So canalicules are coming out of these lacunae spaces, and these canalicules are actually connecting with the other osteocyte or lacunae space, and ultimately they will reach to the central canal. So this is the process through which they are connected to one another to transform the nutrition to one another. So we have lamella. Each lamella contains osteocytes with the lacunae, and each lacunae contains canaliculi which are connecting to the other one. So here we can see the collagen fibers, osteons central canal, perforating canals. Along with that, another thing which is important, this is the most outer layer which is known as periosteum, and this periosteum has a fibrous layer and a cellular layer inside, and they are connected through, or they are just firmly adherent to the underlying compact bone or circumferential lamella with the special type of fibers which are known as perforating fibers or Sharpie's fibers. Another diagram of an osteon, we can see already we have discussed the orientation of fibers to give strength to the osteon. Central canal with the artery, vein and nerve, a lamella, 3 lamella we can see here. We can see the single lacunar space with the osteocyte inside and projections from these canaliculi from the osteocytes and connecting to the surrounding osteocytes to make a good canalicular network. So here we can see the lacunar spaces with osteocytes, along with we see the communication of these canaliculi. So canaliculi of one osteocyte or lacunar space is connecting with the other one. And this is our Haversian system with central canal or Haversian canal and perforating canal, already we discussed. This is another diagram of osteon, central canal, lamella, lacunae, canaliculi. And then on the outer aspect we have interstitial lamella. So in between the two osteons, this space is known as interstitial lamellae, which are again collagen fibers. So here we can also appreciate Haversian canal, canaliculi, lacunae and lamella, which are collagen fibers. That was about the compact bone. Now the spongy bone is also known as cancellous or trabecular, as we can see here that they are arranged in shape of a trabeculae. So the functional unit, in other words, we can say for the compact bone is osteon, but for the spongy bone is trabeculae. Contains trabeculae found at the end of the long bones, ribs, sternum, hips, vertebrae, cranium. They are web-like arrangement, already we discussed, a network-like structure, and we need these bones where low stress areas, where weight of bone would not be a problem, so we need more like a space of we are having this spongy bone in the skull cap, because the presence of this bone is making the weight of the skull lighter, so we need a little bit lighter in weight bone. So Allah subhanahu wa ta'ala has given our spongy bone as a characteristic that is lying in the skull bone and in the other ends of the long bones to make the weight of the bone lighter also. So here we can see this is the trabecular bone, which is spongy bone. This is the compact bone, and if we go inside the compact bone, what we can see, the trabeculae. So trabecular structure is same like osteon, but the major difference we can see here, we don't have any Haversian system over here. Here we can see the Haversian system, a vascular system, but here we don't see any Haversian system. So the spongy bone is devoid of Haversian system. So we enlarge a trabeculae. This trabeculae is enlarged over here. What we can see the same layers of osteoblast on the outer aspect aligned together with the lamellae inside, and these lamellae contains lacunar spaces which contains osteocytes, and again the canaliculi are present there. So canaliculi is there, lacunae is there, osteocytes are there, parallel layer of collagen fibers is there, but we are lacking a central canal, or we are lacking a Haversian system in the spongy bone as compared to. So this is a major difference between a spongy, that we are lacking a Haversian system in spongy as compared to compact. And under microscope it will appear like this, that we have a clumps of compact bone in shape of a trabeculae, and in between them we have a adipose tissue present inside. As we all know that this area is for the formation of bone marrow, so bone marrow is present there. Early life we have red bone marrow, but in later life it is adipose tissue, and adipose tissue basically appears in a shape of a signet ring appearance. So the cells are empty over here, and we can see only the nuclei in the periphery. So this is another enlarged view of the spongy bone, which is showing the marrow cavity, a red marrow, along with the trabeculae, and in between trabeculae we can see the lacunae and osteocytes present there, making the system which is known as Havergen system. Now this is a diagram which is forming a development of an osteon and how these osteoclasts are working actually. So this whole area is the bone, mineralized bone, and if we can see this is the blood vessels area, or in the peripheral side we have osteoblasts. So what happened initially, these are the cells which are osteoclasts, the giant cells we have discussed, and if you can see they have projection and we name them as ruffled border. So just like the drilling material, they are penetrating through this canal, so form a canal inside the bone over here. So they actually started from here and they are tunneling it, and finally they form a tunnel. So the most inner portion is these osteoclast cells. So this part is known as cutting cone over here, you can see. And then we have behind the osteoclast, we have, as we can see here, as this tunnel is formed and these osteoclasts are eating the bone inside, the newly formed osteoblasts are started to penetrate inside that tunnel. So this tunnel periphery is filled by the osteoblast, and as they are going in, they are also secreting the material which is known as osteoid, and as they are going forward and forward, below that this is a reversal zone where this formation is taking place, and finally this whole bone, which is now making a form of a tunnel, will be changed into a newly formed bone, because osteoblasts are forming the new bone. So new calcified bone is formed over here, and the old bone is absorbed by the osteoclasts which are tunneling inside the old bone. Here we can see this is the osteon with the osteoblast. This is a closing osteon, and here we can see resorption cavity is formed. These are the osteoclast cells which are start to make this area or tunneling this area, and they are starting to resorb the surrounding bones, and here you can see forming resorption cavity over here at this place. So this how an osteon is formed, how a tunnel is formed by the help of these osteoclasts, and old edge is eaten by these cells, and from behind the osteoblasts are forming the new bone along with the calcified bone. And this is a diagram which is showing ossification. The term ossification means that how a bone is developing in the body. We have three types of ossification. The first one, which is known as endochondral, also known as cartilaginous ossification. The second type is membranous ossification, and the third type is membranocartilaginous ossification. Here we are discussing endochondral or cartilaginous ossification, that initially when the baby is developing, and the bones which are formed by the cartilaginous ossification, initially we have a cartilage structure initially, and then in that cartilage structure a bony collar is formed in the middle, and then as the bony collar is forming and penetrating inside this cartilage, this blood comes inside and penetrating inside, and as we go ahead we can see the concentration of this cartilage is getting less, and only it is remaining on the periphery of the bones, and in the final we can see that the cartilage remains only on the articular surface where it has to meet with the other bone or at the epiphyseal end. And in the middle, if we can see, we have a central, this is the epiphysis, this is diaphysis, and this is the metaphysis. So this metaphysis also contains epiphyseal growth plate where the cartilage is present, and earlier we have discussed that thickness of the bone depends upon the cells which are present here in the periosteum known as osteoblasts, and they divide and make the thickness of the bone, while the length of the bone or height of the person is depending upon how the cells are dividing over here. They divide mitotically, and then as they are dividing, they are increasing the concentration of cells on bone matrix in the lower part as well as upper part, so they are increasing the height. So our height increase by the division mainly of these cells which are epiphyseal growth plate of cartilage, and ultimately this epiphyseal growth after complete ossification remain as just a single epiphyseal line. and further now in this part of the bone, or when it is developed, no further height will be increased because the bone is completely formed. Now there is no epiphyseal plate over here, only the line is there, which is the remnant of this epiphyseal growth. So we have discussing endochondral ossification. This is the same slide which is forming osteogenesis process of the long bones, that we initially have a hyaline cartilage there, and then in between them there is a degenerative hypertrophic cartilage present over there, and in the periphery we have the hyaline cartilage, and as the bone blood vessels are penetrating inside, they are divided into ascending branches and descending branches, and primary ossification centers develop in the middle and the bone growth takes place. This is the epiphyseal plate of cartilage over here, and along with that, not only the arteries are penetrating into the middle of the diaphysis, they are also penetrating into the epiphyseal ends, which are known as epiphyseal capillaries. So they are connecting together. Along with that, these arteries, which are the arteries present in the middle, they are dividing into ascending branches and descending branches, and finally these arteries, which are known as nutrient artery, they ascending branches are anastomosic with the arteries which are epiphyseal and metaphyseal arteries in the upper part as well as in the lower part. And along with that, if we see, the concentration of the cartilage is decreasing as the bone is increasing, and finally when bone is formed, only the hyaline cartilage is present on the articular surfaces and the rest of the bone is devoid of cartilage now. So there is no further growth in length will be taking place for this specific person. So this is whole about the bone, compact and spongy, with their development. We have only discussed here cartilaginous ossification. For membranous ossification, just to give 2 points, that in membranous ossification we have a layer of membrane of fibrous tissue forms, and then it converted into a bony tissue. So in between there is no cartilage formation there, so there will be no cartilaginous model in the membranous ossification. And the third type is membrano-cartilaginous ossification. In our body there are certain bones which are originating from cartilaginous model as well as membranous model. Mainly these bones are in the bones which are forming in the skull, so that will be discussed later inshallah. So this is all about the bone, spongy and compact bone. So if you need any questions, you can email me and ask the question on this email address. Thank you very much.